Dear students, welcome to my class. Today I am going to discuss genome sequencing projects, which is included in your genomics and proteomics syllabus unit 2. Please consider this class as in continuation to my previous class on sequence assembly. This topic includes genome sequencing projects overall, accessing and retrieving genome project informations from the web, which is in your syllabus, comparative genomics, types of genome sequencing approaches, physical and genetic map. We will also discuss a little bit about Synteny, assembly by scaffolding. Previously, you have understood what is assembly, how a normal assembly is done. But in a big genome sequencing projects, when the genome is very large, suppose wheat, maize, etc., you have to have different steps. One is the scaffolding, assembly by scaffolding. We'll be discussing that also in today's class. And why genomics? Briefly, what gives you what genomics gives you, and how to define it. Basically, high throughput techniques like high throughput sequencing is used for analyzing the structure and function of genes all together. That is genomics, and it's always been associated with some other omics techniques like transcriptomics, proteomics, metabolomics, etc. Until genomic tools were available, molecular analysis of plant expression confined to single gene approach only. Recent technological developments, especially here, the high throughput genome sequencing enabled analysis of organism's genome in terms of genome organization, expression, and interactions. These three things actually are the basis of knowing the whole biology of the organism. Genomics is study of the way genes and genetic informations are organized within the genome, which is one of the very important aspect of genomics. Before that, we were not very concerned about how genes and other elements are arranged in the chromosome. We didn't have much of information. Genomics have subsequently expanded our ability to analyze and understand plant genomes and reduce the gap between genotype and phenotype. Earlier genetics, that's why we call it as a reverse genetics. Now we know the genes and we can predict the phenotype. Earlier, we started with the phenotype and somehow we tried to have some knowledge about the genotype, which was not very precise. Now we start with the genes and the genetic element, then we come back to the phenotype, which is more precise. The general perspectives of genomics, omics techniques, basically, as I told you, omics techniques all are interrelated. We don't have a boundary between genomics and transcriptomics, transcriptomics and proteomics, proteomics and metabolics. We don't have a separate boundary. They are all related. And in present perspectives, if you see genomics, transcriptomics, proteomics, metabolomics, and many other things are coming like phenomics, lipidomics, glycomics, epigenomics, which is again a very important part, new addition. Lots of research is going on about the epigenomics part of expressions. Now, after that, a bioinformatics part comes, which is an integral part of genomics, and through bioinformatics, we now get the access to those data, as well as different types of analysis could be done through bioinformatics, and public domains are available for doing these things. Genome sequencing projects basically aims at discovering all genes and their functions in a particular species. Plant genome sequencing projects initially started with, as the genome sequencing projects used to take long time, and resources were less. High throughput methods confined to Senger's dideoxy methods only. NGS was not that applicable at that time. Machineries were not there, so few model organisms were considered. As you know, some model organisms you can name like E. coli, Saccharomyces cerevisiae, C. elegans, mouse, and in case of plants, Arabidopsis. These were some of the plants and animals and organisms. Included in microbes, plants and animals were the first organisms which were considered for genome sequencing projects. If we come to the plants, plant has got a different organization in terms of its genome other than animals and microbes. It's more complex. Plant genomes are characterized by huge variations in genome size and ploidy. If you can understand, most of the photosynthetic angiosperms have got 10 to 100 times bigger genomes than human. Human is the most developed organism, but these plants have higher number of genes and higher number of genomes in their genetic material. For example, rice, wheat, maize, enormous genome sizes. And moreover, the number of genes also are more in case of these plants rather than human. Human has got only 25 to 30,000 genes, but in case of your rice and all, you will get 68,000, 90,000 or 1 lakh genes. Especially 35% of the plant genes are related to photosynthesis. That's an additional thing which we don't have. So to sequence large genomes, the most important thing is the de novo sequencing. De novo sequencing means you, after having a genetic map, you get a framework map which is not complete. In patches of the genome, you get some sequences so that these sequences can be used as landmarks while assembly of next resequencing. So sequencing is done in 2 stages. First, you get a framework map. Framework sequence, like some parts of the genome you can sequence and align it into chromosomes depending upon the markers present. Now there will be some gaps. These gaps can be mapped using physically by 2 other processes like gap filling and genome walking, which we'll be discussing. Coming back to the technology, again I told you DNA sequencing methods evolved, and it's helped scientists to undertake genome sequencing projects in realistic time scale. As if you can see, human genome sequencing was started in 1992 and it was finished in 2003. That means it took 13 years. In 13 years time, people cannot go for sequencing for all the organisms, but due to the involvement of very sophisticated techniques, evolution of sequencing techniques, now within 1 day the whole genome can be sequenced. It helped in sequencing of genomes. So many more organisms are being almost at a time one time will come when all organisms listed till date will have genome informations. Genomic sequences will be available. The story started with Bacteriophage lambda, which was sequenced in 1983. Before that, actually, lambda 5X174 was the first organism to be sequenced, but that sequence was not that complete, and different method was used for sequencing lambda 5X174. If you take as genome sequencing project, first was the Bacteriophage lambda, which was done in 1983, and next the other sequencing of other organisms started with E. coli, Saccharomyces cerevisiae, Arabidopsis thaliana, which is the model organism for plants, which is actually a weed. It's very much prominent in tropical and subtropical countries, especially tropical countries like America and US and many other countries. Oryza sativa, Cinerotus elegans, fruit fly, mouse, chimpanzee, human, all are followed. Next. Main objectives of plant genome sequencing projects, if you see, two things: obtaining sequences of the genes and the regulatory sequences. And second, which is also very important, is how these sequences can be utilized and get it accessed by other researchers for their scientific research. Rationale of genome sequencing projects, what we have got and why we need to do genome sequencing. If you see, they have given us, these genome sequencing projects gave us few things which were never possible. Number one is the knowing exactly the genome size of an organism. Then the gene numbers, pattern of sequence duplication. See, gene duplication is very much associated with amount of expression. Suppose you want to express the metabolite in a plant. Now you have to see how many copy numbers are there. Copy numbers are there. More copy numbers means they will express more. Catalog of transposable elements, which are very important for manipulation. Synteny relationship means comparative genomics. Like if you know the sequence of grass, then you could take that information to study the genomes of many plants of Gramineae family. All grass, rice, etc., barley, etc., wheat, oats, all are in the family Gramineae. If you have genome information about one Gramineae family member, then that sequence information can be utilized in various ways to study other Gramineae family species. That is called as the comparative genomics. Comparative genomics is based on syntenic relationship. Syntenic relationship, you can see, I will show you in a later part of my presentation. Now these things, genome size, knowing all those things, actually help in few applications like crop improvement. If you want to get a better crop, disease resistant crop, you want to clone a particular gene responsible for particular metabolite synthesis, then you can transfer these genes to another commercial variety. All those things are possible only because of successful completion of many genome sequencing projects in plants. The various steps in genome sequencing projects, first you have to have a genetic map. A genetic map gives you the landmarks. Then you have to do high throughput, high molecular weight DNA extraction. And those things we have already discussed in your previous semester genetic engineering course. Then you prepare a genetic DNA library. Then you go for shotgun sequencing. After shotgun sequencing, your wet lab is finished. You get lots of sequences. Those sequences has to be assembled, which we have discussed in my previous class. Annotation. Next is annotation, means which sequence have got what functions and where it located in the chromosome can be done using bioinformatics methods. One example is BLAST, and there are other things also. Complete physical map construction. After assembly, you get a consensus sequence. Then making these informations available through some databases, which are basically called as genome browsers. In the later part of my presentation, I'll show you some of the genome browsers where you can get all the informations. Like in NCBI homepage, you get lots of databases and lots of tools to do your analysis. You can download things, you can do analysis online. Similarly, a complete genome in a separate portal gives you all those informations like you get it in your NCBI homepage. This is one diagrammatic representation taking hemophilia influenza, one bacteria, as model organism for the whole sequencing project steps. You can see extraction, shotgun sequencing, separation by electrophoresis, then fragment preparation, cloning, preparation, plating, then assembly, end sequencing. If some sequencing is still have some gaps, then it is called as end sequencing. Then construct the contigs. After the contigs are there and the whole genome is organized and graphical representation is present in the browsers. There are two different types of approaches for sequencing. One is by clone approach, which takes more time. You get a framework map, you separate fragments from your genome, you make clones, then separately those clones has to be subcloned and then sequenced, then assembled. It takes time. But there is another method called as whole genome shotgun approach. You mix whole of the genome, all chromosomes together, you sonicate, then you make fragments. Those fragments are actually now sequenced. Using NGS also you can do that, and you get lots of sequences. Then those sequences are actually mapped one by one after the other, which is called as the assembly. Now for this assembly, again a framework map is required, which is called as the scaffolds. I will discuss these things, assembly by scaffolding. It's a huge number of clones, which number of sequences has to be dealt with. That's why we need a framework. Then only this framework can be used as resequencing of the genome, which we will be discussing. Now during this overlaps, you can see here how overlaps are mapped actually. How do you assemble sequences on the basis of overlapping sequences? It is shown here. This is again we got it from your Genome 3 by T.A. Brown. And during assembly, you get sometimes problem because of presence of lots of repetitive elements present in plant organisms, plant species. Plant species especially very much around 60 to 70% of your genome is actually various kinds of repeats in higher eukaryotes. And plants, they have tandem repeats as well as genome-wide repeats. Now, tandem repeats, while assembling, because these repeats are one after the other, one after the other, one after the other, up to 100 times, you sometimes miss some places while assembly. You can see this, the pink part, the first sequence in the pink part and the second sequence in the pink part and the last sequence in the pink part has got the same ZATTA repeats. So first one may get associated with the last one. So in between, in that region, pink region, you will lose while assembly. Same thing may happen in case of your genome-wide repeats, but here in case of genome-wide repeats, there is a chance that you will lose not only repeats, some coding regions also in between repetitive elements. That's a big problem. Now, depending upon your purpose, the whole sequencing methodology is the same, but you have different approaches of sequencing. One is already we have discussed, which is de novo sequencing. De novo sequencing, you start with scratch. You only have a genetic map and you start to get a framework map, which we have discussed earlier, which is scaffolds actually. In part of the genome is getting sequenced. And after that, you go for the whole genome resequencing. Means now you will arrange the whole of the fragments upon those framework scaffolds. Transcriptome sequencing, you know what is that. Now, very significant new advances in genome sequencing project is the epigenetic sequencing. Now, what is epigenetics? Actually, some factors modify our DNA like methylation, acetylation, etc. They don't change our genetic material, but they affect the genetic material and regulate expression. To know these things, we have methods like genomics related to DNA methylation, genomics related to histone modifications. There are methods. There are platforms like MethyLSeq, ChipCheck, and ATAC sequencing technologies which are used for epigenetic sequencing. Targeted resequencing, you can go for specifically targeted resequencing of a particular part of a genome. Genotyping by sequencing, these are methods you can read in details, and if you join any plant biology laboratory later on in your PhD or in higher studies, you will come up with these terms and you have to somehow deal with these problems. What is genotyping by sequencing? What is metagenomics? What is genome editing? I would request you all people to go through in details, and in later part of my classes, in later classes, we will discuss these things: genotyping by sequencing, then metagenomics and genome editing. One of the examples is your CRISPR, which is actually invented for animal genome editing. Now it is being used for plants as well as for other organisms. Now, previously we have discussed assembly in my previous class. Here we are going to discuss little bit about assembly by scaffolding. As I have discussed, you get a framework map, then you go for resequencing. Here in this diagram, I am showing you the A is a genetic map. Before you go for any sequencing, you have to have a genetic map. Now this genetic map is fragmented, as you can see in the B part. These fragments are again shotgun sequenced separately and assembly is done. After its assembly is done, it is mapped in various places which are based on your molecular markers in the genetic map present, which is shown in your C part. Now these are called as the scaffolds. Scaffolds means some part of the genome is being sequenced and they are aligned on the basis of the genetic map, which is part A. Now in the D, if you align, you have some gaps in between. Now these gaps can be filled by a method called as genome walking, which we'll be discussing later on. After these things are is done, this information is actually graphically is uploaded in your genome browser. If you want to verify this, you can go for NCBI genome browser and you just see mouse genome assembly. If you go for mouse genome assembly, you will see there are genetic maps, there are back clones, and there are some other small clones, all which with names of those clones they are being. If you click in any sequence, it will show you in GenBank the whole sequence. Those informations, that's how you actually retrieve your genome sequencing informations nowadays. You need not to download, search the sequences one by one. You can directly go for the graphical representation of the physical map assembly of an organism. You can see some clone names like back something, scar something. If you click on those sequences directly, you can get the back clone in the NCBI GenBank. So from clicking the sequence there in the browser, you can directly go to the database where the sequence is available. Now this leads to some of our topics which we have not discussed here. We'll be discussing these things in after a few classes. What is gap filling? How you do gap filling during your sequencing projects, and genome walking. One of the gap filling method is your genome walking. And for genome walking, you can go for one method called as the PCR method, which is called as nested PCR. Nested PCR. We'll discuss later on. In genomics, in genetic engineering paper, we have discussed little bit about some PCR based techniques. Might be we have discussed this, which is a nested PCR method. Now again, problem created by the repetitive elements. We have to deal with that. There are some kits, there are methods available to deal with those things, which we will be discussing later on. Now a genetic map and a physical map. In case of your genetic map, two things. Genetic maps is based on crossover percentage in a population. So you need a segregating population. These segregating populations may be F2 generation, back crosses, NILs, recombinant inbred lines, double hybrids, etc. Now how big is the population? It will influence the number of crossing over. So we get a estimate of genetic distances between two genes, but this is not exactly the difference in terms of base pairs. Even though genetic map has got some applications, which we will be discussing later on. Identification of quantitative trait loci, one of them. Marker assisted selection, which is very much used by the plant breeding people, because of which you have different brinjal varieties in the market, because of which different bhindi varieties, ladies finger varieties are available in the market, because of which there are different kinds of dals available in the market. It's all because of QTL identification, marker assisted selection. Besides, there are some genetic engineering techniques like map-based cloning, cloning of particular genes based on genetic maps. These are all methods which are dependent upon genetic maps. Then what is a physical map? A physical map is placing of bases one after the other as it exists actually in the chromosomal DNA. For that, you have to do sequencing and assembly. A centimorgan interval may harbor 30 to 100 genes even in a small genome. Therefore, resolution is very low in case of genetic map. To omit this difficulty, we go for physical map. And physical map is obtained by genome sequencing. One method is shotgun sequencing, then assembly. It's very simple. After you have a genetic map is just an estimate of your distances between genes in terms of crossover, number of crossovers in a particular population which is used for mapping. It's based on genetic linkage analysis. But in case of your physical map, we actually know how many base pairs are there in between two genes and exactly how many base pairs the gene contains and exactly how many bases are there for each regulatory sequences. You can see here two diagrams. First, the comparison between genetic map and a physical map. Genetic map has got molecular landmarks, and in case of physical, you have fragments of DNA. And fragments of DNA which might be back probes, ESTs, and many other clones. The different features of genome sequencing research, what we have got by using sequencing. Sequencing newly discovered species, one of the aspects of genome sequencing research. Suppose you have discovered one species and you are not confirmed, and taxonomy people cannot tell you is this a new organism or a new variety of a same species. So how can you determine it? Sequencing or sequence comparison of two organisms can be the solution. By comparing the genome sequence, even a partial genome sequence of a newly discovered species, comparing it with a already existing near organism, you can actually establish whether you have discovered a new species or it's already a variety or a type of a existing species. Research on complex genome plant species like wheat, banana. If you can see banana, there are two types of genomes, A and B form, and in different varieties like Senisompacol, Jahazicol, there are combinations of different genomes. Some are combinations of AAB, some combinations are ABB, means A genome and B genome. And most of the commercial varieties are hybrids of these two. Similarly, you have different levels of ploidies. In case of your wheat, and you have definitely know that there are ploidies, tetraploids, triploids, hexaploids of wheat. So definitely chromosome numbers vary, and chromosome details also vary in case of different types. Those can be analyzed using genomic tools. In aquaculture, sometimes it's very difficult to differentiate between two fish species. One species is not consumed, not interested by the consumer, and one species is very much interested by the consumer, and they cannot be distinguished using very common techniques. So genomics can help you through some markers to differentiate two species of fishes. Large-scale animal sequencing enables rapid livestock improvement. We need more milk, more meat, then genomics can be an application for improving this species. Dolphins are being studied to help in understanding few things in human through comparative biology, and it may help in medicine and conservation also. Now, why model organisms? How a model organism is actually chosen, and on what basis they are regarded as good for us? They should be diploid. There are some characteristics: diploid, appropriate for genetic analysis, amenable to genetic transformation, which is a very important thing, relatively small genome they should have, commonly available tools of resources, means already they should have some resources available. Literature must be available about that organism. We should know in details about that organism. Then only we can call a organism as model organism, and we can start sequencing. Being the focus of research by large community. As you know, Arabidopsis is one of the first plant organisms to be sequenced, which is a weed actually. And why we have taken Arabidopsis as a model crop? It's because of the small genome and its very short life cycle. Within 1.5 months, you can have a complete life cycle of Arabidopsis, and its availability of literature, and moreover its applicability in plant tissue culture. These are the factors which gives edge for Arabidopsis over other plants as model organism. Celera Genomics, one of the Craig Venter's institute, which started sequencing lots of organisms including lotus, legumes, rice, mouse, many other plants, many other bacteria, many other animals, dog, etc. And they have given all these informations in tigr.org. Then there are some specific databases like bean genes for beans, Chlamydomonas, forest 3db, etc. etc. This is the institute, Craig Venter's institute. Craig Venter, if you know, he is the inventor of shotgun sequencing. In his institute, named as TIGR, and actually this is the database for his institute, and it availed many genome sequences of many organisms. Now that institute has merged with two others, and it has become JCVI, the Craig Venter's Institute. Some of the publications about Arabidopsis: some resources for rice. If you can see this page, this is like your NCBI. In the left side, here are data, and below you have BLAST. You can download protein information, you can download functional information, as well as you can get the whole genome assembly. So this is called as the genome gateway, from where you can access all databases related to rice sequence projects. Another example, wheat genome sequencing projects. In wheat, we have sequenced nuclear, chloroplast, and mitochondrial genomes, all, which is helpful in your crop improvement, breeding, gene manipulation, and also helped in phylogenetic studies. Reason for sequencing mitochondrial and chloroplast genome. In some interviews, people ask, why go for mitochondrial and chloroplast sequencing? Now, mitochondrial and chloroplast genes, 70% of them already copied in the normal nuclear genome. So then the question arises, why to go for mitochondrial and chloroplast genome sequencing? Actually, many of the very significant plant expressions are related to mitochondrial and chloroplast genome. One example is your cytoplasmic male sterility, which is very important while getting a new variety during your crop improvement in plants for breeders. Comparative genomics, as I have discussed already, a method to unravel relationship between genomes by describing conserved chromosomes, which is identical, and characterization of syntenic relationship between related species. It helps in providing information about one species, unknown species, in relation to another. Now, what is synteny? Synteny means, suppose there is a chromosome number 2 of one species and chromosome number 4 of another species. You have one genetic map available for chromosome number 2 of one species, where you locate this TA37, GAA, SCAR, etc. These are the molecular markers present in that way. And after genetic analysis, comparison, synteny, you have found that in another related species, chromosome number 4 have got the same sequence of markers present, but they have in different resolutions. So we can say that this second part, chromosome number 4 of another species, actually somehow related to the chromosome number 2 of the first species through evolution. So we can say that these two have syntenic relationship. Whole genome resequencing projects. In case of Arabidopsis, it was started in 2008, where 1001 strains of Arabidopsis in different varieties of Arabidopsis found in different places had to be sequenced. A method was devised which could actually sequence the whole genome in one day, and it helped a lot for the genomics community. Similarly, 50 accessions of wild rice from different places of the world could be sequenced, and now they are available for research. What are the web-based resources available for data retrieval and utilization from genome sequencing projects? There are sequence databases, data mining tools. One example is your BLAST, then phylogenetic analysis tools, etc. Bioinformatics platforms can be useful in analyzing genome sequencing data. Genome gateways, as I have just shown you, Rise Genome Research Gateway. There are mirror sites and genome browsers. Now, genome browsers, how they look like. There are two types of genome browsers. One type of genome browsers which is called as a general genome browsers, through which you can find information for many genome sequencing projects. Many species informations are available. In certain cases, you have only one species. Like example is rice map, which is related to the physical map of rice only. But in case of your ensemble and UCSC genome browser, you will get informations on many species. This is an example of NCBI genome browser. You can see here different tools, different downloadables, different options by which you can do online assembly, online blast, on download sequences through FTP. You can go for blast and you can find out literature also. All things available in your this genome browser. You can go for organism specific browsing also. You can find out organisms. Suppose here custom resources, genome, human genome is separate, microbes, all microbes are put into one, organelles, all organelles put into one, or viruses, prokaryotes separately, and protein informations. Then UCSC genome browser, see coronavirus information is also there. Genome browsers, all these databases are available. This diagram actually summarizes the whole thing about genome sequencing projects. In A, if you can see, there are sequencing of different organisms as it's shown in pictures. Then you start building a database. First you go for NCBI genome, you submit sequences there, then you get publications in PubMeds, then you get other sequence informations, then metabolomics information, transcriptomics informations, proteomics informations all come together. Then in B part, if you can see, we start doing some analysis internally. Suppose NCBI is doing it. They have separated, categorized the data in different databases. Then you arrange the database using a MySQL, or you actually get a database, complete database. Then you create a web interface through which the public user can access this data. It's a very good diagram to show the whole thing about genomics and how you can retrieve and analyze genomics data. Students, I would like to discuss few other things. One thing is you should know linkage and crossover, which you have studied during BSc, and I would request you to go through it and make a note of it. Then different types of repeat elements. We have already studied these things in genetic engineering paper. We'll be discussing these things later on, but still I would like to give you an assignment so that you can read what are the repetitive elements present in organisms. Thank you.